All right. Okay, pleasant good afternoon, everyone. So today we'll be looking at tissues and the integument, relating, of course, to the integumentary system. When we think about tissues and the integumentary system, what tissues come to mind? Connective, epithelial, muscle, and nervous. A nice mnemonic, or in terms of how we could remember it, using these 4 letters, as some of the persons in the class so rightly intimated. Semen is one, or men-C. So C-M-E-N or M-E-N-C. Those letters will point you in the direction of the 4 types of tissue. So let's start with the epithelial. With the epithelial, we have these simple squamous. So these are usually found lining things, ducts, and a location within the glomerulus of kidneys. You look at more in terms of the glomerulus and SNF2, and the alveoli in the lung. Also found at the inner lining of capillaries, right? So they line, and these are the simple. So when you think about simple squamous, just think about lining. They usually found the lining, and they're in one layer. The next one then, if they're not in one layer, they are layered. So you have stratified squamous, and their function is protection, and they're located in the skin, mouth, esophagus, anus, and vagina. All right, so they also belong to the integumentary system, the digestive system in terms of going from mouth to anus, and also part of the reproductive system in terms of the vagina, in terms of the lining there. Now, if you will notice in terms of the stratified squamous, what could you say? If you see a picture of this, what comments could you make about it? You know, if you're looking at it, what would you say? Well, you could see in terms of the it's neat, orderly arrangement. And why I point this out to you is just to let you know, well, later on, when you do look at cancerous cells or cancer tumors, there's a protocol associated with the laying down of cells. So yes, these cells do get sloughed off, and they are replaced by mitosis. Mitosis, of course, is a cloning type of division which identical cells are produced. But the main thing is there's a protocol that says, hey, you know, we're laying down next to one another, and we're laying down, you know, in layers. When you look at cancer cells, and this is one of the things in terms of identification of cells from a histological or perspective, cancerous cells, they don't look nice and flat. These cells are usually confused. They lie on top of one another, which is why they form tumors or bumps. So you don't have this nice layering. This layering pattern is not there. So that is one of the ways, you know, from a histological perspective, when they're looking at tissue, let's say a tissue specimen under a microscope, that's how they could tell it's actually cancerous, because it doesn't have this nice arrangement, but it is instead confused. So what do you mean by confused? Point taken. I'll look to get a sample for next day, right? So I'll be able to show it to you. All right. Yes, go ahead.
So like a skin tag, right? Like a mole. So that be it would be like that, like
a cancerous cell. I am guessing. Yeah. I'm guessing I'm going out on thin ice there. I would say, yeah, that sounds logical, because it is not normal. So it's abnormal. And one of the first things usually broken in terms of protocol is the whole notion of this layering, this neat layering arrangement. Now everybody jumping up on everybody, and you get this bump. Yeah, so as skin tags, I would assume that. I'll look to get some samples of those things. I'm going to take note of it. Hang on to your wagon wheel. I'll write that down. Where is it? My good notes. This is SNF1. Get histologic sample skin tag. That's 1, and 2, cancer. Okay, great. All right. Well, that's a good point you raised there in terms of skin tags. We looked at simple squamous, stratified, which is the layered ones. Now we're looking at simple cuboidal. Now, why do you think they call cuboidal cells cuboidal cells, first of all? When you look at the cells, well, they have a type of, go ahead.
Because of their shape, sir?
Correct is right. Thank you, Crystal. Yeah, because of their shape. We could force a little experimentation in terms of stretching your mind a little bit, but it's a cube. The shape of the cells are cuboidal in shape. So the epithelium, the layering of the cells, these cuboidal shaped cells. And in terms of their location, you also find them in tubules of the kidneys, glands, and ducts. So again, the simple cuboidal, these are found in these areas and they line them. So it's more of a protective function. Final, we have the stratified. Now we looked previously at the simple squamous epithelium. Now we're looking at the stratified cuboidal epithelium. So these are associated with secretion and their location are the larger glands and ducts, example, mammary glands. So in the mammary glands, what do you think they will secrete?
Breast milk.
Yeah, milk. Yeah, breast milk, right? And what hormone is associated with the drawdown of milk?
Prolactin, I think. Prolactin.
Prolactin. What does oxytocin do? What does oxytocin do?
So, and they call that the cuddle hormone or something like that.
Well, oxytocin is
field hormone.
the field hormone.
Oxytocin, that is when you, and something to do with pregnancy, you would have
Yeah,
oxytocin
correct.
and contraction.
Correct, contraction,
Something to do with
uterine
pregnancy,
contraction. Yeah,
Yeah.
uterine contraction indeed. But oxytocin in terms of breast, it also has a function in the breast as it relates to milk.
Never heard of it. I don't know.
It's for the drawdown. So prolactin actually causes the formation of milk, but the drawdown. So in terms of the breast milk that is flowing out of the ducts, down to the baby, that is under the control of oxytocin. And it makes the milk that is already in the breast flow for the current feed and helps the baby to get the milk. Right? So it starts working when a mother expects to feed as well as when the baby starts suckling. So oxytocin is associated with the drawdown of the milk from the duct. So it's present there, but for it to come out, oxytocin, just like how, for uterine contraction, it causes the squeezing of the uterus to get the little bundle of joy out. Similarly, the milk that is present in the mammary glands, in terms of passing through the ducts, that oxytocin does have that squeezing as well to get it out. So whenever you think about oxytocin, think about squeeze, like, you always have somebody who like to squeeze you, an aunt or somebody who like to grab you and squeeze you, right? So think about oxytocin in that regard.
Excuse.
Go ahead, yes?
So when you have a baby, right, and you're breastfeeding and say the milk start to leak, is because oxytocin is released to cause that spraying and leaking and stuff like that.
Yeah, and where you have to wear pads so you don't embarrass yourself. Yeah, I remember my sister when she had my nephew. Yeah, she always used to have to wear the breast pads. Notwithstanding that, she also used a breast pump. In terms of taking the milk out and storing it, right? Because she was producing a lot of milk. Hence the reason why, she was always lactating. Back to the question again, sorry. What was the question again, if you don't mind repeating it?
So I was asking if the oxytocin does cause the release and
yeah, correct.
Okay.
So in terms of that, the drawdown of the milk, yeah, that is responsible. So, and it's also, oxytocin is also stimulated when the baby suckling, when it's drawing on the nipple. That also causes the release of oxytocin, and also that assists in the drawdown of the milk as well. Yeah, so the suckling action also helps with that. But there's some people, they just produce more milk than others, and they do lactate more. And sometimes if they do happen to forget, it could be a little ethical faux pas, it will, until somebody pointed out, excuse me, you're a little wet, in the chest area there. Oops, I forgot to put the pads on. Yeah, so that's a good point you raised there. Thanks. Okay, so then we have stratified. We go to simple columnan. Again, based on shape, the columnar cells are shown here. They are longer than they are wide, hence the reason that they are called columns, right? So they're longer than they are wide, so they're columnar. And these simpler columnar epithelium, they function for absorption, secretion, and protection. and they are found, of course, in the stomach, the small intestines, and the uterus. Very interesting. You know, the small intestines are actually longer than the large intestines, yes? But they call them the small intestines. How much longer is the small intestines than the large intestine? That's a good one. Anybody wants a gander? How long is your, well, your small intestine is actually about 6, let me see, what's the average height of a, okay, I think the average height of a woman is safe to say is 5 feet, or is that on the short side? Let's just say
5'6", not 5'6" is average.
five is kind of short for a woman, 5 feet?
Yeah, I think 5'6" is average for a woman.
5'6", okay, 5'6" in the
Yeah.
middle there, okay. Okay, let's just say a short, just for the maths. Let's just, and you know, 5'6", 5'4", 6'4" is 24, 5. Actually, that would still work. So just imagine your small intestine is about 4, if you were to put 4 women, well, 3 women, you know, on top of one another, it's like 4 women long. That's the length of your small intestine. All right, so anything about, you know, your gut, your small intestine, it's about 4 women long. It's very long, 22 feet. All right. And what about your large intestine? Well, your large intestine, as we just mentioned, is actually shorter than the small intestine. And the large one is only about 1 woman long. So when you're thinking about those two relationships, always remember that the small intestine is 4 times as long, 4 times as long as the large intestine in terms of its length, and in terms of the, well, how long, you're 4 times as long. Just think about the average length of a woman, let's say 5'6", so approximately that. Round it off and say 5 feet. All right, so do remember, small intestine 4 times as long as the large, and the large intestine approximately 5 feet long. Yeah, rather interesting. So the simple columnar associated in location in the stomach and the small intestines, and their functions, they're functional, whereas the others were, you know, the major one was protection. These have special functions, namely absorption and secretion associated with them. All right, let's go on. So now these pseudo stratified, they so-called them pseudo means like faults. So they want to look like, when it's purely stratified, you know, there are layers. Pseudo stratified, you have different, the height. So if you just look at the nuclei, even though it's, you know, it's on a curve here, if it was all straight, all the nuclei would line up, you know, they'd be about the same height. But pseudo stratified means some of them, they are different heights. So they're not all on the same level, they are different heights. So hence the name pseudo-stratified. And these pseudo-stratified, they serve one function primarily, and that is one of secretion. Right? So they do secrete mucus, and they also move things along with the cilia associated with them. And this is, of course, in the respiratory tract. Right? So this one is taken from the respiratory tract. Transitional. Anytime you hear the word transitional, the first word that should come to mind is bladder. Right? You know, it's like a hand and glove. When you think about a hand, you think about a glove. When you hear the word transitional, you should think bladder immediately, because that's one of the primary areas where you would find transitional epithelium. Can anybody think of a way you could correlate those two, transitional with bladder? Anybody could see a necessary link you could make with those two words, transition and bladder?
The bladder extend.
Yes. Oh, so transition as in change. So it transitions from its flat, empty state to the balloon type, or the large structure that is, let's say, associated with that. I like how you're thinking. I like that. Yeah. Anybody have anything else?
So what I mean to join the words?
Oh, no, like how you could remember it. So let's say a question comes, where would you find transitional epithelium? So, you know, if you could draw a mental connection. So your colleague
like a balloon filled with water and release any water.
Right, right. so the transition would be changing, let's say, from a flat balloon. So the word transition means change. Yeah, transitioning, from one state to another, you're changing from one to another. So you can think about it when you think about transitional, yeah, your bladder filling. So normally it's flat. Like what they used to call them back in the day is ice bag. Those bags what they used to use, what they used to fill it with? Not hot water, but the ice bag. I'm not sure if they have them anymore. You fill it with ice. So you can think about it, when you fill it up, it changes its shape. Whereas when it's flat, when it doesn't have the ice, it's flat. So the transitional epithelium, right? Do remember, from the time you hear the word transitional, first word should come into your mind, bladder. Now let me ask you this. Let's just say you drink a whole lot of water and juice and so on, and you walk in. Could you suddenly hear poto and urine all over the place? Could that ever happen?
No, sir.
No? You sure? Okay, well, no, it can't happen. And the reason why, of course, right, we do have, always remember, all our organs, it's covered by a sheath, right? It does have this connective tissue all over it, for anybody here is hunt?
Oh, sir, but my husband do.
Right. So when they skin in the animal, after they take off the fur, you'll see, that's one of the ways you'll notice it. You'll see it has this total connective tissue covering all over, before you actually get down to the meat, and that prevents things like poto and things from exploding. Not going to happen. Notwithstanding that, of course, there are the other organs around it and so on in terms of the bladder. So always remember that you wouldn't just walk down, if you drink too much and poto. What you would do, of course, you put strain on your sphincters. Right? You have the sphincters on the bladder itself, and that could cause strain. And if you put strain on those sphincters, which are really rings of muscle, later in life it could cause what? A word that begins with I, and it leads to
incontinence.
Thank you very much. Yeah, it could lead to incontinence. So which is why, when you do have children, I remember like when I was younger, some people used to say, like a little child, and you go out with them and they say, Mommy, I want you to tell it. And you know what they would say? Come on, be a man, hold it in. Yeah, that's not a very good idea. I mean, to each his own in terms of the rearing of your children. I wouldn't tell you how to rear your children. But, if let's say, a youngster is asking you to use the bathroom, it's a very good idea to let them out to find a place, where he or she could find some relief because of the fact that later in life, yeah, incontinence could happen if it is you put too much strain on those bladder sphincter muscles. Okay? All right, let's go.
Excuse me.
Yeah, go ahead.
I know the transitional epithelium, you tell us to think about the bladder, right?
Correct.
But with the stomach, right? The stomach itself changes shape and all of that too, right?
Correct.
It's the same.
Do you have transitional epithelium in your stomach? To the best of my recollection, the answer is no. No. So it is unique to the bladder itself in terms of transitional epithelium. But that's a very good question because they're very similar in terms of that distension. But there's something that is unique to the bladder, specifically to the bladder, the transitional epithelium. Yeah. So yeah, so do keep that one in mind. I'm glad you raised that point. Yeah. Okay. Let's go forward. So connective tissue now. You have fluid, liquid connective tissue, incidentally blood. Right? Blood, of course, is a fluid connective, liquid connective tissue. And the reason for this is when you look at the embryological development of the body, blood actually develops along with the connective tissue. That's why it's classified as a connective tissue, but of course it's liquid, so they call it a liquid connective tissue. But this has to do with the embryology or the development in utero, of that within those 9 months when the development of the fetus is occurring. So the function, of course, transportation, and it does form part of the cardiovascular system. So this is liquid connective. Let's look at loose connective tissue. And how, if you're looking at loose connective tissue, here we areolar tissue. And where would we find areolar tissue? Well, it functions, it surrounds and supports organ. Areolar tissue is found below the epithelia, subcutaneous layer, example, skin, between muscle and around capillaries. right? So it's a packing tissue, right? In terms of the loose connective, and just by appearance, if you look at it, these are the nuclei, these darkened areas here. These are the nuclei. These are the limits of the cytoplasm. This is as the cell membrane, the outer cell membranes. But if you notice, it's loose. You know, you could see a lot of space in between it, hence the name loose connective. This is another one, of course, in terms of loose connective, adipose tissue. And adipose, when you hear the word adipose, what three-letter word comes to mind?
Fat.
No, fat, right? Yeah, adipose tissue, right? It does. For anybody, do we have anybody here who cooks? We have any chefs here? And in the cooking world, what do they say about fat? Fat is flavor. Is that true in terms of cooking?
So they say.
It
Yeah.
depends on the fat. It depends on the fat too.
Depends on the fat. It have some fat sweeter than some.
Well, pork, you get the best flavor from pork.
Oh yes, yeah, indeed, and all due respect to members of the Muslim faith, but quite so, it does. It pork and bacon and so on. Yes, it does give a delicious flavor. When you look at the French cooking, I was watching this show they were showing you back in the day, in those culinary schools. They actually, next to the stove, they would have a metal jug, and they would have what they say goose fat is the best of the lot. I don't know, I never had it, but they say goose. When you render down the fat off of a goose, they say that is the sweetest of the lot. Yes. So what they would have a metal jug, right next to the stove, because at room temperature it coagulates, it gets solid. So the metal jug will draw the heat from the stove, so it will keep it liquid. And they would cook with goose fat. I understand, like if you cook with goose, if you fry things in it, is absolutely delicious, right? Notwithstanding pork, yes, indeed. Pork is one of the few things where you can actually just show a little bit of salt on it, and it's good to go. All the other meats here earlier, some of them here earlier, to put seasoning in them. Yeah, but pork in and of itself is, it's unique in that regard. Okay. All right, let's get back to this. Adipose tissue. Now this is adipose tissue, and these are the adipocytes, or the fat cells as they're called. Now, if you would notice, usually when we look at cells, you see it's pinkish because they use a particular stain, a stain called a H&E, hematoxylin and eosin. Do we have to know that? No, not really. All you have to know, when you see pink is a H&E stain. And what's the difference in terms of the stain absorbance? What would you say between, if you were to look at this, we're looking at some areolar tissue and adipose tissue. What could you say about the absorbance of the stain when you look at areola compared to adipose? What could you say?
Because adipose is more fatty, it did not absorb as much stain as the areola.
Correct is right, yeah. So in general, yeah, the adipocytes fat, just the nature of it, it doesn't absorb. So whereas here, the areola, it absorbs the stain, the H&E stain, fat doesn't. It's only, you could see just in the periphery along the membranes, it absorbs it. So usually when you see a sample, just as this, from the time you see things that look in, what does this look like? If you saw this, what does this remind you of? This image.
They look like marshmallows.
Marshmallows, right? So if you see a marshmallow, or what else it could look like? Okay. Tiles. It looked like tiles on the ground, yeah. So if it looked like marshmallow or tiles, from the time you see it, right, it's adipose tissue. Now I'm saying that, why do I say this? Oh yeah, when you have a spotter coming up eventually, the assessment. And yeah, I can tell you, especially with this one on tissue, yes, you'll be well placed to be able to identify the tissue by sight, because a significant part of the assessment will just come, what is this? What is this? Asking you to label by sight the different types of tissue. So one of the ways, especially with adipose, always think about this marshmallow, right? Because it is filled with fat. And the fat resists the stain. It doesn't take it up at all. And that's why it remains white. Let's get to dense connective. So the dense connective, these functions to attach things, and they resist tensile strength. And where do you find them? In your tendons and your ligaments. Ligaments, bone to bone or bone to muscle?
Bone to muscle. No, so ligaments, bone to bone, sorry.
Bone to bone, that's right. And tendons?
Bone to muscle.
Bone to muscle, very good, right? What's a nice way so you wouldn't get confused? Anybody? Because it's easy to get confused. You know, you're saying, I ain't gonna forget that easy. But you know, there's this old saying, when I used to play football, I remember the coach always used to tell us, pressure does do what? Pressure does bust?
Pipe.
Pressure does bust pipe. I don't know if you ever heard that before. No? Yeah, but when you're under pressure, you know, yeah, things go wrong. So how could we know tendons, that ligaments is bone to bone and tendons is muscle to bone? Anybody could think of a way where we could draw that connection? So ligament, bone to bone, how could we draw that connection between the word ligament and bone? So I just learn it. Me know about no connection. Okay, fair enough. So you just learn it. In terms of the tendon, what is the largest tendon in your body? And it's found close to your heel.
Is that the Achilles?
Correct is right, the Achilles tendon. And there's a story behind it. You know, back in the day when this is from Greek mythology. Anybody knows the story? Anyway,
yes, sir.
Go ahead, let me hear you.
I have an idea.
So I can't remember the story, but I know it was to do with Achilles, and when they cut his heel, because he was a great fighter, and that was the only way they got to disable him.
Right. And why that was his weak spot? There's a reason why there and only there was his weak spot. Anybody know? It have to do with the river Styx. The river and him doing good, so they wanted to make him virtually immortal. Okay. All right. So back in the day, you know, the Greek gods, what they used to do, when you do really good things, you know, they would reward you. And he did some good things. So what they did, they dipped him in the river Styx to make him immortal. But to dip him in, then they had to hold him somewhere. And where they held him was, you guessed it, by his tendon there, and they dip him in the river. Right. So that is why there was his only weak spot, because that's where they held him to dip him in the river. That's how the story goes. So that was his one weak spot. All right, so you could feel it, your ligament, your Achilles, the tendon, the Achilles tendon. That's where they held him to dip him into the river Styx. So that's the story, and that's why, you know, that was his one weak spot. But in truth and in fact, if you ever do get your tendon cut, you wouldn't be able to walk. You know, dorsiflexion doesn't occur. Your foot would not be able to move, right? So literally, you have to drag yourself along. All right. So do remember that tendon and ligaments. How I usually remember ligaments is that, you know, in your knee. I don't know, for those of us who follow football or even basketball, we have any ballers here? No? Anybody who's follow football?
Cricket.
Cricket. Okay, cricket. And I'm not sure this injury happens very often, but you might have heard it in sports in general. ACL. You know, I don't know if you ever heard, like, you know, some people in the knee, you know, they damage ACL, right? Which is the anterior cruciate ligament. And when you think about your knee, you know, to me, when you think about knee, it's one set of bone inside of there, right? So the anterior cruciate ligament is that ligament. That's how I normally relate bone to bone, because when I think about the knee, I know it's one set of bone, whereas down by a tendon, well, there's a muscle. Which muscle is that one? Your gastrocnemius, or your calf muscle, right? That is the one that the ligament, the tendon, attaches to your calcaneus or your heel, right? The calcaneal bone, that is where it attaches to it. That's the point of attachment, right? So your Achilles tendon, the muscle is your calf muscle or your calcaneus muscle to your heel, right? Your gastrocnemius muscle to your calcaneus bone. Right, so that is the attachment and association with the Achilles, whereas in your knee, it's bones. And that's how, you know, I relate to the fact that ligaments, the ACL injuries associated there. Here's some dense connective. We have irregular, and they're so called because if you notice, they're looking like some kind of piece of cloth, yes? So it have some dresses, they look like satin, and they do tingling, you know, they do that kind of thing on it. My mother is a seamstress, that's how I know about these things, you know. They look. Yes. So it looking like a, you know, it have a kind of thing they do with dresses
I
and so
just
on.
remember it because it has kind of a marble look.
A marble appearance. That is true. Very true. Right. So when you see this one, do remember it's dense, irregular. And again, it's not, you know, nice and smooth and layered. It does look irregular. Yeah, it does indeed look irregular. And withstand tension and location is in the capsules of the organs and joints and the dermis of the skin. This one is unique. Notice a different color, right? It's purple in terms of the stain used. And from the time you see these little circles, looking like little, what? It look like eyes. What does this look like? If you see it, what comes to mind immediately? So that looking like the Caribbean islands. I see in St. Vincent, St. Kitts, Trinidad and Tobago down there. Fair enough. You could say it looks like islands in the Caribbean Sea. So when you see this, you know, this pattern, you could say, from the time you see it, purple with these white is known as highland cartilage. What else does it look like? What does it look like to anybody else when you see it?
Chicken pox.
Chicken pox. There you go. On your skin. Yeah, I like the parallel, right? So chicken pox, islands in the Caribbean, or just white. From the time you see them, you know it's hyaline. And the location is the larynx, trachea, the nasal septum, and the ends of the bone. Where's your nasal septum? Where's your nasal septum?
Your nose? Your nose?
Is it nose, yeah? And what part?
Yes.
What part of the...
Separating your nostril. That
Yeah.
line that's separating your nostril.
Correct. And some people, yeah, they do pierce it, right?
Yes.
You know, and my understanding is particularly with cocaine usage, what it does, it burns a hole in that septum, you know, such that you could see right across. So let's say if you go out on a, let's say a blind date, as it were, and the significant other you're with, you know, you just look and you see a hole, you know, you could look straight across from one nose hole to another, unless he or she has a good, you know, could show you some baby pictures, which just shows that they had it for life. Well, you know, you might want to be thinking along the line, maybe this person might be using some kind of, you know, a hard drug, namely along in the cocaine family, right? So that's a thought. Do remember that it does burn through the septum. Cocaine in particular, one of the drugs that does that. Yeah, go ahead.
The highland cartilage, this image here, so that you start to pay attention to the purple color.
Yes.
Right? They're using a different stain.
Correct. Let me see if I could get that stain for you. As opposed to these. So
these will always appear in any exam, whatever.
Yeah.
Will appear color.
Yeah, you see these get a little pinkish. You get a pinkish coloration, but this one is purple. It's a different stain. Let's see.
Okay.
Stain. Let's see if I could get stain for hyaline cartilage. Is it needed for? It's something called safranin O. Right? Is a kind of a stain. Do you need to know that? It's not needed for the course. Right? But just be aware of the fact that the reason why it's different in color is a different stain. Right, so they use safranin O. So that clearly means then that if they do use the normal stain, which is H&E, it doesn't work. You know, if they use it, you wouldn't see the differentiation, and you don't see this the distinction made. So you know that's how they do it. So when they try it and it don't work, what do? Try another stain, and they tried another stain and it worked. Hey, so we'll go ahead and continue to use the safranin O as it relates to hyaline. But that is not on the syllabus in terms of knowledge of the stains. No, it's not on the syllabus. But just be aware that they do use, when you see different colors, it's because some are taken up and some don't. So that's why they use different types of stain. Good question. All right, so here it is elastic cartilage now. This is very flexible. And where do you find it? In the pinna and your epiglottis. Where is your epiglottis? I forget, where's your epiglottis? Where would you find it?
That flap that is separate the
flap, correct.
trachea from the
esophagus. Yeah, me too. I always like to say, very good. The epiglottis is like I was going to say policeman, but it's not. Mark, when I use the word man, it's gender neutral, so don't think I'm being biased in any way. I'll say police person. But you know, it's like a policeman or a traffic light, the epiglottis. What would happen if the epiglottis doesn't work? what would happen? And you look to eat something. What would happen? You're eating a meal, epiglottis not working.
You choke?
You could choke. Food could go down in your lungs. Yeah, I see what you're saying. And ultimately, the gag reflex associated with choking will be initiated, right? As you know, when things, as your colleague just said, things will go down in your lungs, because what the epiglottis does is like a policeman in that regard. It guides things. So when you're talking, it covers up the esophagus, so air could pass through the trachea, and then when you're eating, it covers the trachea, so things go down into the esophagus, into your stomach. So if it's not working, well, that's a big problem, because when you're eating, normally the trachea is covered up, so that flap is not there, things could go down inside of there, and that's a problem. Why it's a problem when food gets down into your lungs? One word begins with I.
Infection.
Oh yeah, and that's a big problem. That's why you have this gag or the cough reflex when things go down into your lungs. Yeah, when you're eating, as the old people say, and it gone down the wrong throat. Yeah, you begin to cough, and the reason you're coughing, you're getting it out. That is really important. Yeah, all right. So your epiglottis and your pinna is part of your.
I'm going to go with air.
Yeah, it's part of your air, the pinna. And what part of the air is the pinna?
The cartilage part, the one on top. Yeah.
Part on top here.
Yeah. So again, some people is boring, not the normal boring part, but the higher, the cartilaginous part, the part above it. So not the lobe of the ear, but above that. Yeah, that cartilage. Indeed, that's your pinna. So whenever you think of this, do remember hard connective tissue in terms of cartilage. These are two of the locations. Next one, we're going to look at fibrocartilage. Again, different stain. Fibro, tensile strength, shock absorber. And you find these where? Where would you need shock absorbance? Intervertebral discs and your pubic symphysis. Pubic symphysis, that is found, close to your pubic bone, right? But the intervertebral disc is very important, and that's where you have this hard connective or tissue or cartilage. It acts as a shock absorber. That is why what we could do, I remember when I was in primary school, we had this flight of stairs. So what do we used to do as children? We used to see how much stairs you could climb and jump all the way down. Anybody used to do that in school? So you start on 1 and you jump down, you climb up 2 and you jump 2, you 3 and you jump all the way down 4. Of course the adventurous ones would hit about 7. And really they say.
That again, no, sir?
They say that.
You jump and remain right there.
And they say God protects fools and children. I don't know if you ever hear that. Yeah, yes. But it's very true. Or the dim-witted and children. And it's very true. Because now that I'm thinking back on that, look at how many things could have gone wrong.
Yes, you could have break something like.
You understand? You understand? But back then, you're thinking about that, you're just jumping and you're having a good time. So
children fearless.
Quite so. Yeah. And in terms of birth, I think the only fears that we're actually born with is the fear of falling and the fear of loud noises. That is the only two. You ever realize, like with a baby, when you clap your hands loud, you'll see them, open up their eyes.
Yeah, they stiffen up on
they stiffen, they have that fear. Do they have a fear of falling? I'm not sure. But I know definitely fear of loud noises, that is one you're born with. But with falling, I'm just go walking, trying to walk at least, and but that's the only fears. All the other fears are actually learned along the way. I do know indeed fear of loud noises. That is something that is from the get-go. They have that as a reflex action. Structural, this is osseous tissue, also known as bone. So again, the stain what they use here, this is something that is used to clean cuts. Anybody want to guess what kind of stain it is? It looks very similar in color. That they put it on cuts. It begins with I.
Iodine?
Correct is right, yeah. So this is the only, whenever you see, and this is a circles, it's bone. From the time you see these concentric circles and this brown color, everything looking like potholes, right? Right away. From the time you see these circles, is bone. And the brown color in particular. I can't think of any other stain that is used specifically with iodine. I think bone is about the only one. So it's unique. When you see the brown color, immediately you could think about bone. What else does this look
It
like?
looks like wood too. Like when you cut
Correct.
the tree and you see the ring.
Yes,
It looks like
yes.
that.
Yeah, so that's another way. Yeah, indeed it does. Yeah, a tree, you cut it. So you see the rings, so you can think about it that way as well. Yeah. Anything else it looks like to anybody?
Kind of like an octopus. I mean,
An octopus. Yeah, yeah. I like how you're thinking. An octopus up close. Yeah, it sure does.
Sometimes, like when I do tie-dye, sir.
I like how you think. Yes, and if you tie this area so it get dark, and then when you release it, you get the patterning.
All of my stuff, I am related to like fabrics.
What you saying?
Remembering it.
Yeah, I ain't mad at all
So
with
the
you.
one with Highland, I put it as like a cheetah print. A cheetah print thing. So if I think of everything as fabrics, it's easier to remember for me at least.
I like how you're thinking. I ain't mad at you. That's very good.
You could post those fabrics in a group too, because I like that too.
Yeah. And you have a web page in terms of, so you must post a web page in the group as well. Yeah, in that regard, but it does look indeed like tie-dye. Yeah. All right, so now we're moving. We're almost there. We have nine slides again. Muscle tissue. So when we think about muscle, we have skeletal muscle tissue, and skeletal muscle in terms of what attachment. So when you think about skeletal muscle is muscular system. Let me ask you a very simple question. Why do they call skeletal muscle, by the way? For a very simple reason. Because it is attached to?
It's attached to the skeleton.
Yeah, that's why they call it skeletal muscle, right? It's attached. And it brings about movement due to the interaction of which other organ systems? Two others. So you usually have the muscle. Well, actually, there's two right here. One more.
Yeah, muscular and skeletal.
Yes, muscular skeleton. And there's one more that causes muscle contraction. Which one causes muscle contraction now?
Nervous?
The nervous system, very good. Yeah. So whenever you hear the word movement, immediately in your mind, and three things, from the time you hear movement, in your mind, you're thinking skeletal, muscle, and nervous system. So the nervous system provides the impulse. When muscles get an impulse, they only have one movement, they contract. When you remove the impulse, they go back to their normal length, right? And because of that attachment, from muscle to bone is, what is that? Tendon, right? So when you do have that attachment, that's how, of course, you have some joint or pivot around which the movement occurs, but because it's shortened, it pulls on it, and that's how you have the bone moving, and that's how movement occurs. But skeletal muscle, they're long, they're striated, I'm going the wrong direction. It has these stripes associated with them. You can notice on the surface, they have these stripes, and they're multinucleated. So it has many nuclei, not just one. And of course, their function is for movement and generate heat. Could you tell me what is a reflex action that we have where we have muscle contraction that generates heat? When does that happen? When you say muscle contract continuously, pardon? Shivering, thank you very much. Yeah. Like if you're going somewhere very cold, and you start to shiver, the reason why you're shivering is muscle contraction. One of the waste products when you break down glucose, right? So when you're looking at the muscle tissue, remember in the blood, what does the blood do? Let's say you eat your meal. So the digestive system is traversed by, of course, a whole network or plexus of blood vessels. So the nutrients get into the blood and they're taken to the different cells. When they go to the cells, what do the cells do? Well, they break it down. If in particular, if it's sugar, it's derivative. That's the main one that is broken down. So it gets into the cells, and these nutrients, right? What do? Break it down, and when the nutrients are broken down, it generates ATP. So if you're looking at glucose or food, food plus oxygen gives you carbon dioxide, water, and energy. when you're looking at skeletal muscle in particular, when it's getting those impulses, because it needs the impulses to contract, notwithstanding the impulses coming in, but it needs energy to do it, one of the byproducts is heat. I think we could identify with that those who actually, let's say, when you're doing any type of movement, not only do you sweat, right, which is a cooling mechanism, but you get hot, you find your temperature goes up. So for shivering, that's a reflex mechanism that generates heat. When your body recognizes the core temperature is beginning to go too low, well, it would start to shiver. And what is our core temperature?
36.
Right, our core temperature. Well, 37 is a good average. It goes between 36 and a half to 38 and a half. Yeah, 36.5 to 38.5. So 37 is a good average, right, in terms of your body temperature, your core temperature. So when it realizes it's going too low, it would initiate that shivering reflex, and some people, the teeth chatter, but what your body is doing, that contraction is actually making heat. So we go from, yeah, go ahead.
This muscle, right, this skeletal muscle. So this is the only one that multinucleated, right? Or it have others that multinucleated. Because the smooth muscle, they have one nucleus, and the
cardiac muscle has one, one.
And it tapered, right?
Yeah, and the smooth.
Yeah.
Yes, that's correct.
When I'm looking at this, right, because I was kind of confused about, you see where you see any white lines separating them and they're looking like, well, like long sausages, right? So that the length itself is the muscle, right? And so each one of them have more than one nucleus in it.
Correct. So hence the reason why they call it multinucleated. Whereas the others, they only have one within, within you could see the cell membrane, and within each cell membrane, you only have one nucleus. Whereas with these, within one, well, you're not seeing them here, but within one cell, you have many nuclei. And the reason why you have so many nuclei is because of the fact, well, it have a lot of contraction. It does a specific job of contracting. It has to do it all the time. So therefore, it needs to have a lot more control. So hence the reason it has more nuclei as compared to the others.
Okay, so but it long then, because like I'm going back to the example of the
yes,
the tapered muscles. You see how you could see like a beginning and an end of it under the microscope?
I understand.
In all of the images, these just long. So I was just trying to figure out if
let me write that down. Yeah, let me write that down for next day. I'll bring some. Yeah, because this is a kind of sample.
Because you see like how the cardiac muscles, they have the intercalated this, and you can see where they separate, and this one is an individual and that one is an individual.
Multinucleation.
I was trying to figure out like where these join, or like that.
Understand. And I just never thought about it. I just took that as gospel truth. But I understand you want to see it physically. You want to see where it ending. You want to see it within one cell membrane, right? I'd get some images for you. That's a very good question. I just took it as gospel truth, right? I never questioned it. Yeah, there's a longitudinal section you're looking at. But yes, good. That's a good point. So with this one, as your colleague quite rightly says, it does have these intercalated discs. And the intercalated discs, they're very important for what reason? What do they do? One word, really, begins with C, and it have to do with electricity, and it does the same thing what copper does in a electric wire.
Conduction.
Yeah, correct, excellent. So what the intercalated discs do, they ensure that conduction occurs throughout cardiac tissue. And why is that important? If you don't have that electrical impulse traveling through to all the cells, cardiac cells within the heart, what would happen to the heart? It wouldn't. Now, usually when you think about the heart, you have a direct contraction and relaxation of the atria and ventricles. So therefore, all the cells have to get in the atria, they have to get the impulse at the same time. And there's a slight delay that the ventricles get. So you have that alternating, contraction and relaxation. If it didn't get that, what would happen? Well, the heart would go into something that is known as fibrillation. So it just had a quiver. And to stop it from quivering or to restart it, now the impulse for the heart is initiated at the SA node, right, which is the sinoatrial node. How I remember it, when I think about rhythm, you know, associated with the heart, when I think about rhythm, I think about drums. When I think about drums, I think about Africa. When I think about Africa, one part of Africa is what? South Africa. Yeah, that's a Nelson Mandela from, right? So SA, South Africa, that's how, you know, the rhythm. So the impulse for the heart is generated by the SA node, right, which I call the South African node, but really stands for sinoatrial. So please don't go and put South African node in your paper. I will laugh at it. I will give you 0, right? It's the sinoatrial node, also known as the SA node, right? So that's where the impulse. So all of these, the intercalated discs, they facilitate the conduction of the electrical impulse. If it doesn't happen, you'd have fibrillation. And how do you stop fibrillation? There's a specific device they use to stop fibrillation. What is it called? Mm-hmm. Huh?
AED or defibrillator.
Ooh, automatic, are you talking the automatic defibrillators? In fact, quite right. There are some now, very good, particularly by the Philips Corporation, that actually tells you where to put, you know, the electrodes. And it tells you the electrodes are in the right place, and then it tells you clear, and then it will, you know, it talks to you, this device, and it actually tells you, okay, place it on the device itself, on the electrodes. It shows you where to place it, and you just have to put it in the general area, and it'll tell you if it's in the correct area or not. And then when it's in the correct area, it'll say okay, and then it'll say clear. So a defibrillator, what it does, it stops fibrillation or the shaking of the heart. The heart begins to quiver. So intercalated disc, very important for conduction. Then you have the smooth visceral muscle. These are usually found where? Anytime you hear the word smooth muscle, what comes to mind?
Organs.
The hollow organs. Or
The
which?
alimentary canal.
Alimentary, yeah, actually that was the one I was looking for. I was going to say what organ system is most closely associated with smooth muscle. So if it's the alimentary canal, the digestive, excellent, well done, right? So the digestive system, right, is associated with smooth or visceral muscle. And let me see if I remember, how long is the large and the small intestines? Which one is longer?
Small intestine.
Small intestines.
How long is the small intestine? Approximately.
About the length of 4 average.
22 feet. 22 feet.
Yes. 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 So now for this one though, if let's say I ask that question and you put approximately 4 women, I would give you it right. Right. But if you do have any standardized test, please don't put that, I don't know how they would mark, but if you put it for me, I would give you that one right. It's approximately 4 women long, right? And the large intestine is approximately how long? About 5 feet. Yeah, about one, like about 1, yeah. So since it's 4 times as long. So it's between 5 and 5.5 feet long. Yeah. Okay, very good. So the smooth muscle, is that under control of the autonomic nervous system? So in other words, do you have any control over the contraction or the movement of food through your gut?
No, it's involuntary.
It's involuntary, so it's under the autonomic control, right? Very good. So smooth muscle, those contractions, very important for moving the food from mouth, well, not really mouth, because from esophagus all the way down to anus. That's very true. The visceral control, smooth muscle. So last thing we're going to look at, right, nervous tissue. The structure of a nervous tissue. What is a new nervous tissue called? Sorry, let me rephrase that. A nervous cell. A nerve cell is called a neuron. Very good. Yeah, very good. A neuron. So this is it here. So this is the cell body. So when normally we look at a cell, the cell body, so this is the cell membrane. What is the structure inside of here? Well, actually all here. These are the nucleolus, a nucleus, very good. And then you have these long projections, right? The dendrites, they bring impulses into the cell body, and you have the axons, which take the impulses away. one of the interesting things with the nervous tissue or neuron is the only tissue that could do what? A neuron. It has one particular function that nothing else, no other cell could do. And it has to do with electrical impulses. Not only could it transmit it, but it could also, it begins with G.
Generate.
Oh yeah. So when you look at a neuron, in this region here is known as the axon hillock. Yeah, because it looks like a small hill. And in this region of the axon hillock, what do you find? Well, you find sodium channels. You have a high concentration of sodium channels. And what's so important about sodium channels? Yeah. Now, the inside, if you were to think on the other side of the membrane, so you're looking at the outside. So imagine there's a door. You open the door and you're pushing your hand and you're touching the wall. Yeah, inside the cell. What charge is located on the inside of a cell of a nervous tissue? Normally, what's the charge on the inside? Is it negative or positive?
Positive.
The inside, is it negative or positive? Right. Well, okay, it was a 50-50 and actually negative is the answer. So I'm mad at you, right? So it's negative. Inside is negative. So what happens now, if sodium gates open, so you have these channels or like doors, and there's a very high concentration in the axon hillock, very high. And there's a specific reason for that. So when it reaches threshold, or you have these impulses coming from other cells along the dendrites, it causes these doors to open, sodium gates. Now, all around the neuron, you have sodium floating, in the milieu, whatever substance is bathing it. So the sodium now, when it sees these doors open, what do you think it will do? The door open, sodium outside and a sodium door open. It rushes in, moves in, all right? So it goes inside the cell. Now, sodium, when you think about sodium, the sodium ion, what charge does it have associated with it? Negative or positive?
Positive.
A positive charge. Remember, inside is negative. So positive rushes in. And when you have a large number of sodium ion rushing in, the inside of the membrane, remember there's like opening a door and touching the wall, on the inside of a room. So it's the same thing here. Think about the room as being this whole neuron. You have these sodium gates or doors. The doors open, sodium rushes in. Inside is negative, but because it's positive, all of these positive charges causes a change in polarity on the inside from negative to positive. And that change from inside negative to inside positive is known as depolarization. Yeah? So, yeah?
Sorry.
Go ahead.
So I was gonna ask, like in active transport, right? You learn about the sodium potassium pump. This is the same pump, or this pump is just for sodium?
Oh, no, this one is specific. Yeah. So with the sodium potassium, which is 3-2 in terms of the exchange, right? But this one is very specific for sodium. So sodium alone, and is only specific, is very specific for neurons, and is very specific. The high concentration of these gates, you have them all over the cell, yes, you'll have sodium gates, but specifically in this area known as the axon hillock, which is just before the area where it forms the axon, you have a high concentration. Sodium rushes in, causes depolarization. And what happens? So now inside is positive, but the area right down here is negative. What happens when you have a positive and a negative charge? It pulls the charge, it attracts it. And that's how you have this wave of positivity. It begins to move down the axon, right? But what initiates it is that a whole lot of sodium rushing in, and that is initiated because you have these impulses coming from the other. So these dendrites here, they're attached to other nerve cells. And when it reaches a certain level or threshold, that is what causes these gates to fly open. When they open, sodium rushes in, depolarization, inside is positive, and the charge now begins to move down the axon. Because remember, it's like a tunnel, and all the way down the tunnel is negative. You have this positivity here now, and it's pulled along. It's pulled because of the force of attraction, and it moves down.
So it will only let in the sodium, and then the gates would close and nothing else comes in.
Right. Yes, after a certain time it does close. Once the that threshold period is passed, yeah, it does close. Bam. So when you're looking at the action potential, it has a waveform. There's a part where, yes, the gates open, and after threshold is reached, the gates close, sodium gates close. Right? I don't have it here, but if you could revisit it, you'll see specifically on the graph, when you look at generation of an action potential, there's a part where these gates close. Whoops. Yeah, they do close, shut, right? And the charge moves down because of the attraction, and it moves down. Then, of course, you have the nodes of Ranvier, which is a little gaps. And the thing with the nodes of Ranvier, at the nodes of Ranvier, you also have sodium gates. And it boosts what it does, it boosts the charge coming along. Because remember, the charge is coming along. As it passes those gates, which are located in the node of Ranvier, they open and sodium rushes in. So then, it gives it like a boost. And that is why, very importantly, in the nodes of Ranvier, where you don't have the myelin sheath wrapped around the axon, what you have happening there is you have a lot of sodium gates. In a little space where you don't have the wrapping of the myelin sheath, yeah, you have sodium gates, and that pulls it along. Rather interesting, I tell you. And all of that is so incredible. It's happening in your body all the time. It's just incredible. All right. Last thing I want to mention then is the neuroglia, or these are the supportive cells, also known as glue, right? And these serve as support for the neurons themselves. All right. Any questions? All right. So
Could you repeat what you just said, please?
Oh, sure. So the last set of cells is the neuroglia. So what they serve as, these are support cells for the neurons. They actually help them to ensure that they do their function properly, the neuroglia. So the word neuroglia actually means glue, neuron glue, right? So these are usually found in close proximity to the nervous tissue, and they act as support for the neurons. Okay. All right. Yes, go ahead.
You were sticking just now, and then my whole